So it's really nice to talk about research we've been doing for the past 3 years. And for this presentation, I put together results and graphics from several presentations and papers. So from this point of view, I also excuse myself for the slides sometimes if they're not fully polished. So to start with, I wanted to tell a bit about our group, also to the wider Finnish bioinformatics community. So as Elio mentioned, I lead the graph algorithms and bioinformatics group. This is part of now the algorithmic bioinformatics laboratory in the Department of Computer Science. And we like to do research both on the theory side on graph algorithms, but also on the really applied side on software for genomic and sequencing data. And I think the best the most satisfying sort of research happens at the intersection of the two areas. In our laboratory, we have other researchers, Leena Salmela, Simon Pulisi, Veli Mäkinen, and Juha Kärkkäinen, working on various topics in algorithm bioinformatics, from string algorithms to other applications, for example, in pangenomics. I prepared the slide hopefully at a very high level, assuming a wider audience. If you have any more technical questions, I'm happy to take them after the talk. And now to explain the problem of what does it mean to decompose a graph or a weighted graph, I would like to give a very brief overview of the core problem of decomposing a network flow. So you could think of this kind of a setting where you have some production facilities on the left, and then you have some, for example, some stores on the right, and these production facilities on the left produce a quantity of some object. And this object must make its way through a network, for example, here, and must arrive to the stores in some required quantities. But now this is just a flow of objects, and we would like to actually transport these objects to the stores. So in practice, we might think we want to actually order a number of trucks that would actually take the objects from the factory to the shops. So we could decompose these objects along, for example, this kind of five routes with one truck per route that takes a certain amount of this product and brings it to a specified store. And in this example, these routes are drawn as color paths, and there are 5 paths of them. So if you wish, for this toy example, we would need 5 trucks to carry these objects from the factories to the stores. But you might think, well, we don't want 5 trucks. We would like a fewer number of trucks if possible. And in this case, we could actually route these objects with 3 trucks only. So we could have one truck along the red route, one along the green route, and one along the orange route. And now, if you know a bit more about computer science, this problem of finding the minimum number of paths to decompose a network flow is an NP-hard problem. So it is very likely that it doesn't admit an efficient algorithm in theory that works efficiently for any given input. So why did I give this overview of kind of products being produced and being transported to stores along this kind of network flow? What is the connection to bioinformatics? Well, apparently there's a very nice connection in my point of view. You can think of these routes. There's 3 routes that are drawn here with different colors as different genomic sequences. And then these genomic sequences touch at those areas that are shared among them. So for example, by sharing, we could mean having a common substrate, identical string labeling that shared area. So here we would have GCC in both sequences, and here we would have TG in both sequences. Why is this? What is the decomposition problem? Well, in some applications, we don't have the paths, but we need to recover the paths from this kind of a mixed data. So our data would be just very much like a flow. We would have some structure, so sort of like network structure in terms of a graph. We would have some weights on the graph saying how much, so what is the abundance of the genomic sequence on that part of the graph, and in addition, what is the string labeling that part of the graph. And well, notice there was this initial decomposition that I showed, and we want to decompose this flow into the correct genomic sequences. So if you were to decompose this network flow in this manner, this would be a wrong decomposition. It wouldn't correspond to the ground truth that we're trying to discover. And following the Occam's razor principle, you could think, well, the smallest explanation of the data is the one we prefer. And let's just work with this kind of a sort of notion for the rest of this talk. Now, let's talk about a specific application where this kind of a multi-assembly problem can be applied. So this is the problem of RNA transcript discovery and quantification. So what it means is assembling the RNA transcripts expressed in a cell or in a tissue from RNA sequencing reads. So as you probably know, the central dogma in molecular biology is that from DNA, information is transcribed to RNA. Then in complex organisms such as human, through alternative splicing, such RNA creates different isoform, different mRNA molecules. So for example, this one has all exons in it, but some molecules could skip some exons. So for example, this one skips the third exon, and this one skips the fourth exon. And studying these mRNA molecules is important because then we can understand what proteins are translated in the cell. Now, this is how biology, like a simplified view of biology. Now, sequencing helps. How does sequencing enter into this picture, and how does it lead to a decomposition problem? Now, let's do another drawing of this setting. We have a genome, and we have a gene in it with 4 exons. And through transcription and alternative splicing, we obtain 3 different transcripts with different expression levels. So in this example, 20, 10, and 30. And they all have different sets of exons. Now, through RNA sequencing, we are sequencing all these transcripts together. So we obtain reads from each one of them, but all these reads are mixed together. Now, these reads we could, to construct such a weighted graph, we align them to the genome. We have reads fully aligning only inside exons, and then we have reads aligning across exons, so skipping the introns. And for example, this read here indicates that these 2 exons have been observed as consecutive in some RNA transcript. And this read here tells that the exon 1 and exon 3 have been observed as consecutive in one RNA transcript. Now, based on these exons and these sort of read spanning 2 exons, we can construct a graph. So we can put the exons as nodes. And whenever we have such a read overlapping 2 exons, we put an edge directed from left to right. And now we have a weighted graph, which we need to decompose into weighted paths to ideally recover the RNA transcripts from which these reads were sequenced. So if these are the ground truth transcripts, which we don't know and we need to find, these would correspond to this kind of paths in the graph, which we now need to find. Okay, but this is very much an ideal scenario where data is perfect. There's no sort of wrong numbers, no wrong alignments. The sequencing coverage is uniform and whatnot. But in real data, this doesn't work. This doesn't happen. So we have if we have 3 exons here, then use a perfect numbers here in theory. But then in practice, you have this kind of graph where we do not have the paths, where we do not have the transcripts, and we need to recover the transcripts. The numbers are a bit off because the sequencing coverage is not uniform. We have issues with read alignments, with constructing the graph, identifying the right exon boundaries. On the other hand, we might also have additional information. So if we have long reads, we might also have information that, for example, exon 1 has been observed as followed by exon 3 and exon 5 in some transcript because there's a long read aligning across all 3 exons. So on the one hand, there's more noise in the data. On the other hand, there's more features from the data that we can exploit in this decomposition problem. Now, I hope I set up the problem. Now let's see how to solve this problem. Well, as I mentioned, this problem in theory, it's NP-hard, so it's very likely it doesn't admit an efficient algorithm. But for many NP-hard problems admit efficient solvers via a paradigm called integer linear programming. that is often fast on moderately sized inputs. And very briefly, how this works is that this kind of integer linear program is made up of variables. They can be binary, so 0, 1. They can have integer values, or they can have real values. And then we have constraints on these variables. So meaning inequalities on linear combinations of these variables. So for example, we can do sum of sets of variables, or we can also multiply these variables by a constant and sum them up. And then we can impose inequalities on this kind of linear combinations of variables. But you don't need to know very much about the technicalities about this kind of formalism unless you actually develop solvers for this kind of decomposition problems. And in fact, many RNA transcript assembly tools are based on this paradigm, and there are many. One big issue that they have is that they need some candidate paths that they need to start with. So you need to provide them some candidate transcripts, and then they will find the right set of transcripts with the right abundances that best correspond to the data. But as you see, this is already a heuristic step. How do we choose these candidate paths? On the other hand, you could just also enumerate all possible paths in the graph. But in theory, there are exponentially many. So this would lead to a huge linear integer program that then wouldn't be fast to solve. So already this kind of setting means, oh, there's something that doesn't work. So why is this paradigm not working for this problem? So we had work with Fernando, Lucia, and Brendan showing that actually we can skip this kind of candidate path initial phase. We can encode the search for paths in the integer linear program itself. So now getting a bit more into technical details, we can, if we want to formulate k transcripts that we need to find, we can introduce a set of sort of a binary variable for every edge and for every path index that we need to find. And then for path I, then we will have this kind of binary variables. And then we want that whenever a binary variable is set to 1, that encodes an edge selected in the path. So now if you look at this red path here, so this is a path from a source to a sink, as we would like. And these binary variables have value 1 only on the edges of the path and have value 0 otherwise. And we can add additional constraints that this happens exactly on the edges of the path. You don't need to worry about that. But the important part is that we eliminated the need of candidate paths to start with, because now the integer linear program will also need to find the right values to these binary variables to induce a path. But we delegate this search for candidate paths to the integer linear programming solver itself. We don't need to have this kind of initial step where we give either some candidate paths or exponentially many paths. And once we have variables and constraints to model these kind of k paths, then we can say, well, we want that these paths actually decompose the flow. So we want that the flow value, so the abundance of every edge, let's call it FUV, which is a constant given in input for every edge, equals the weight of the path I multiplied by the times whether this path goes through the edge UV or not. And we sum this for all paths that could potentially go through the edge UV. So on the right, we have the weight that we need to explain with solution paths, so the way that we need to decompose. And on the left, we have actually the weight that from the solution paths that decomposes that edge actually in the solution. And we want these two quantities to be the same for every edge. So we want this kind of weighted paths, we want them to completely decompose the given flow values. Now, I hope that, so once we can include the search for paths in the integer linear program, then we can also solve other decomposition models. So there's another model called inexact flow decomposition, where for every edge, we don't have a single value that we need to exactly decompose, but we have two values, a lower bound and an upper bound, like an interval, where this kind of weighted solution paths must fall. So the sum of the weights of the paths going through the edge in the solution must be in this interval for every edge. So this means that every edge gets a range, like a confidence sort of not in the probabilistic sense, but a confidence interval, and we want the weighted paths to fall in these intervals for every edge. There's also a decomposition model called least square decomposition, where we don't add this kind of constraints, but then we penalize with the error in the decomposition. So for every edge, we look at the input, weighted got, flow valued got, we subtract what we actually decomposed, and we square this difference, and this is an error of an edge, and we sum up all these errors. So ideally, if there's a perfect decomposition, all these errors are zero, so then there's no error, and then all the flow is perfectly decomposed. If not, we want this kind of weighted paths when we overlay them to fit as close as possible to the given weights, flow values, inexact flow values. There's also another model where these kind of errors, we're not kind of squaring them up to penalize them in the objective function, but we want them smaller than sort of also some kind of slack in the path. But it's not important to actually understand what this model is. But what's important to say is that once we can include the search for paths in the LP model itself, then all these models can be efficiently solved. And let's see what I mean by that. So here we have a data set on sort of human reference, gene reference, and we group these into sort of the size of the minimum solution, minimum flow decomposition. Here is the number of graphs in each of these class. This was a previous algorithm which was like exact and sort of exponential time. For this kind of large graph, it solved about half of the inputs in 54 seconds, for example, on average here. This was a heuristic algorithm that runs on all inputs. On the largest graphs, it's half a minute, but then it starts to have a difference with respect to what it should actually have on average. So it actually gets 4 more paths on average than it should get. But now if we do this kind of ILP models, we solve all instances, we solve them optimally, and we can, in some cases, we can even be faster than this kind of heuristic decomposition algorithms, and definitely faster than this kind of previous exponential time algorithms. So in this kind of larger instances, we have like a three times speed up with respect to this other previous exact algorithm. Now, let's see a possible optimization we could do to this kind of integer linear programs. So the idea is to help the integer linear programming solver in sort of reducing the search space for solutions. And we can identify something that we call safe paths to limit, sort of to fix some variables, some binary variables to 1 already when we pass this program to the solver. And let's look at this edge. So this edge must be decomposed. And what is the way to get to this edge? Well, to get to this edge, there's only one way to get here. So any path that decomposes this edge must come from here. And once it decomposes this edge, must continue here because there's a unique edge going out. And here there's also a unique edge going out. And now we have a fork, so we cannot be sure where this kind of solution path goes. But we're sure that once we decompose this edge, we must have the full blue path. And now, same for this edge. The only way to get here is to come through here. So this kind of yellow, sort of orange path also must appear in some solution. And the same for these other 2 paths. But now there's an interesting property of these 4 paths. Once the solution path takes 1, it cannot come back to take another one. So for example, from the blue path, we cannot reach back to the violet path. There's no reachability from the end of this to the beginning of the violet path. And the other way around, from the end of the violet path, we cannot go back to the beginning of the blue path. And in fact, all these 4 paths here, color paths, are pairwise unreachable. So we know they must be used by different solution paths. And with this information, we can fix some binary variables. So we can assign the blue path, for example, to the first solution path without loss of generality, the orange path to the second solution path, and so on until the violet path to the fourth solution path. And helping the solver in this manner, we can speed up the original integer linear program by even 30 times. So this is a more difficult data set. On average, we solve in like over 1 minute. The larger graphs, but now we can do it instead of 1 minute, we can do it in 3 seconds on average. Okay, but this was for these kind of exact decompositions. But what about this kind of inputs with errors? Well, the inputs with errors are even more interesting because the average speed up now for the larger instances becomes even 100 times on this kind of one data set, sort of one decomposition model, this minimum path error. And for the least squares can become even 1,000 times. So instead of 100 seconds, we do it in about 0.16 seconds. Okay, so putting it all together, I hope that now we have really scalable methods for multi-assembly problems. So when we have an input like this, is it this decomposition or this decomposition? What are the true sequences that generate this kind of mixed sequencing data? And let me sort of, so all these the big picture was that we can formulate k-paths. With it, we can add additional variables and constraints to solve all these kind of different models. And then we can optimize this kind of finding the k-paths by setting some binary variables. And now we had all this work was in different papers and different projects. So now we want to actually put all of this together. So we want to make so we are working on a Python package that puts all these models together with all optimizations. That is easy to use. So you just do a pip install flow paths, and it will install all requirements, and then you can just create your solver and you can just solve it for all kind of decomposition models. And then because of the architecture of this package, then all kinds of optimization would be inherited by all possible decomposition models. We are also working on the documentation, so we hope this is really, really like easy to use and people start using this when developing new multi-assembly tools. So to sum up, decomposing a weighted graph is a basic primitive in multi-assembly problems. It's an NP-hard problem, but it's solvable efficiently via integer linear programming. So we can avoid generating candidate paths, and we can be flexible and support many decomposition models. It's solvable in seconds with all these kind of optimizations, and even 1,000 times faster than naively. And we have this kind of, we're working on this kind of flow paths Python package. And with this, I'd like to conclude and acknowledge the many people that worked on these sort of projects and the funding from the Research Council of Finland and the ERC. Yeah. Thank you very much. I'm happy to take questions.
Yeah, thank you very much for the fascinating talk, Alexandru. If somebody has a question about the presentation or Alexandru, this would be the moment.
So, do you hear me?
Yes.
Yes.
Okay. Thanks a lot, Alexandru. It's a very interesting problem. And yeah, first I would like to clarify something. If I understand you correctly, you made comparison to other methods. Is this you mainly talk about the computation time? Is there any comparison to the accuracy about this your algorithm?
Yeah, that's a very good question. So,
Okay.
So in this sort of work, or in this, at least in these presentations, we focused on speeding up speed up optimizations. Because data is not perfect, and I would expect that even these kind of least squares and mean path errors, they are at the core of these tools that I cited, but these tools actually model other things.
Okay.
Yeah, so we wanted to optimize the core thing to make a flexible solution on which you can add constraints on top, and then, but actually to have the groundwork to have something fast, and then when you develop a new multi-assembly tool, then you add other constraints.
Okay, so another question which is probably a bit general. So this is for mRNAs, obviously, and then we know very well where the exons are very well defined. But we also know that currently we have those non-coding RNAs, so where they also have isophones, but probably they don't have very clear structure as compared to mRNAs. So have you, or is there someone else who's doing this research in this field, in non-coding R field, by using a similar approach to identify the possible isophones quickly and accurately?
Yeah, I'm not knowledgeable in that.
Mm,
Here, working on mRNAs, and we also assume a reference, so we can align to the genome and the graphs become acyclic. If you have even the de novo setting, the graphs may not have cycles in general.
mm,
At least what we implemented so far assumes doesn't assume cycles. So, but these things can be generalized to cycles, but probably it's not fully trivial.
okay.
On the other hand, this interlinear programming, they're used a lot in the industry, like for vehicle routing, for like power grid optimization, and then this kind of, it's easy to model, for example, not trusting edges or saying, because then the ILP model will just choose, should I use this edge or not? And we delegate this to the ILP model. So I do believe it's flexible enough.
Okay, thank you.
Yeah, thank you.
So, Alexandru, do you envision this approach being widely adopted in large-scale RNA-seq studies?
Yeah, that's a very good question. So at least I'm very happy about this package because now we put everything together and it's easy to install. On the other hand, and it's fast. So on the other hand, there's people need to know about it and actually learn a bit. I mean, understand a bit how it works and how it can be used. So it's not easy. But maybe if we teach it to our students and something like that, then